Great.
Hi guys. Hope that you are all doing well. So I, just like last week, I will email you when I get the handout posted for tomorrow's lab. Also, if you look on Moodle, kind of at the top of near the top of the Moodle page, I wrote out my current guesstimate of what topics we're going to do on each of the remaining days, because again, we're just continuing behindness. So I kind of wrote out, and there's not that many days left, so I just wrote what topic I thought I was going to get to on each day. If that has to change, we'll see. So today we're going to kind of finish talking about a variety of things related to autoimmunity. And one of those is the final piece of this clonal deviation and clonal suppression example that I started talking about last time. So last time I told you about this issue of clonal deviation or clonal suppression, where this is all really related to the idea that naive T cells particularly naive helper T cells, can be pushed in different directions when they are activated towards Th1 versus Th2, versus Treg versus Th17, etc. And that in some cases, one of those particular responses might be protective or keep you from having pathology, while others may actually lead to some host damage or pathology. And you saw the celiac example of deviation. I've shown you a little bit about leishmania and talked a little bit about some details with Helminth infection. I talked a little bit about the data that is emerging on Helminth infection and SARS-CoV-2. So I gave you a few examples of this last time. But I actually left out sort of the most famous story because it takes a while and I didn't have enough time last time, so I decided to put it now. And I also will tell you that this is a section that I have definitely updated this year because I think that our understanding of this issue has really been changing a bit over time, so I tried to make it up to date. So a lot of this is related to Helminth infection. Helminths are parasitic worms. They include things like roundworms, flukes, and tapeworms, or nematodes, trematodes, and cestodes. And you can see some examples of diseases that are caused by these parasitic worms here. So one of them is a tapeworm infection, one is schistosomiasis, also things like trichinosis, onchocerciasis, hookworm, elephantiasis, ascariasis, all of those things. And so these are all parasite infections with multicellular parasitic worms, or I don't think they're all, I think there might be one that's unicellular, but whatever. And so these are a really big problem, and one thing I want you to know about this is that if we think about the history of infectious disease, people have probably been getting infected with these microbes since before there were people. So some of the sort of pre-human hominids were probably being infected with these organisms. There is probably no situation where there were ever people who did not have Helminth infection. Because, of course, these are so important in our sort of history, we have a really good immune response that has evolved to deal with them, and that's largely the Th2 response. And so you can see a mast cell on the left, and notice all of the granules in that mast cell, both in this microscopy image and in this cartoon. Remember that mast cells have Fc epsilon receptors on their surface, and are able to basically bind up all the IgE antibody. And then if an antigen is present that binds to that IgE antibody, that cross-links the mast cell and leads to the mast cell expelling its granules. So the granules are pre-made. That's going to become something that's actually important on Friday. The granules are pre-made, and we can have basically these granule components immediately released. And the idea here is that you might imagine that this might be something that is good for dealing with a microbe that is perhaps too big to phagocytose. We're not going to be able to phagocytose and kill inside a cell. So instead, we're going to secrete toxic products to try to destroy that microbe. You can see the textbook showing the same thing at the top here with, in this case, eosinophils. It can be a few different cell types degranulating to try to attack a schistosome, one of those parasites I told you about. And again, here you see a schistosome larvae, and here you see eosinophils. So you can see the eosinophils are never going to be able to phagocytose this great big larvae. And instead, they are secreting these toxic products at this schistosome larvae. So like I said, we have this great response that has evolved to help us with Helminth infection, and we have been evolving with helminths pretty much since before there were humans. And we know that if we make a Th2 response to Helminth infection, as I've told you before, we tend to get these protective effects, and so you've seen this slide a few times at this point. But if something were to happen where someone were to make a Th1 response, that Th1 response would not be helpful against the helminth, so it would not actually do anything about that microbe, and it would instead actually cause some host damage. And so it's in fact quite important that we are correctly making the Th2 response and sort of making the balanced response here to deal with this infection. So in thinking about this, I want you to think a little bit about different aspects of your life. And I want you to think about the question, have you ever spent much of your time or really seriously thought about whether or not you had a Helminth infection? Have you ever been worried about having a Helminth infection? Have you ever thought of, has this ever really been a thing that's been a big deal in your life? So Jonah says no, Brenna says no, Juliana says no.
Personally, yes.
Personally, okay, you have?
I grew up with OCD, so.
You grew up what?
With OCD, so I was
Okay.
terrified of everything.
Okay. Huh?
That's an irrational fear.
Irrational fear, okay.
I would say it was rational.
Okay, any other thoughts about
I'm
this?
saying mine were.
So,
I was freaked out.
the piece of information that sort of ties into all of this, and again, I'm going to present this in sort of the classic way that we've been thinking about it for a long time, and then I'm going to show you kind of how our understanding of this has changed a little bit. So know that I'm going to kind of tweak this process as we go through it. Is that we know that Helminth infection right now is largely only seen in some areas of the world, and that's shown on the right. So generally, Helminth infections are happening in areas that are in red in this image. That's where people tend to get Helminth infections, and a lot of it is due to sort of public health details and sort of issues with public health. And you can see that, so one of the reasons why you might not have thought a lot about getting Helminth infection is because perhaps you look at this and you're like, oh, I live in a place where there's very low Helminth infection. What has been noticed statistically, and I'm going to be telling you a little bit about in the next few slides some kind of statistical things, officially they are all epidemiological observations, is that if you look at where people in the world have autoimmune diseases, they are in the areas in red on the left-hand map. And so you can see autoimmune disease incidence, high, medium, low, and you can see Helminth infection, high, medium, low, in these two world maps. And so what do you notice when you look at those two maps? Yeah, Shelby.
They're opposites.
They're opposites. It seems as though in places where people get Helminth infection, they don't have autoimmune disorders. Or in places where people do have autoimmune disorders, they don't get Helminth infection. And so this was sort of something that people noticed. They started kind of wondering about, thinking about. Remember, I showed you that SARS-CoV-2 paper, which came out in 2024, maybe 2023, talking about how people who have Helminth infections have totally different responses to SARS-CoV-2 because their immune response is pushed towards a Th2 response. This idea was sort of originally referred to as the hygiene hypothesis, and some other observations really extended the hygiene hypothesis. And so again, I want to point out that the things I'm going to show you on the next couple of slides are epidemiological observations. We can hypothesize or speculate what the mechanism is, what change in immune systems is leading to this, but right now I'm just showing you like data about things that helped us make a hypothesis. And so one other part of the hygiene hypothesis that people realized is that we tended to see, and now you'll see some of these slides talk about allergy instead of autoimmunity, but it depending on how you see this, sometimes it's allergy, sometimes it's autoimmunity, that we tend to see more of these allergies or autoimmune disorders in westernized countries. We tend to see, you actually can see an epidemiological link that you are more likely to have allergy or autoimmune, particularly allergies, if you are from a family with fewer kids. If you have more siblings, you're more likely to not have allergies. If you're from sort of a more affluent urban home, you're more likely to have allergies. And if you sort of are in an area with sort of good sanitation, low Helminth burden. While alternatively, you are unlikely to have allergies if you're in a developing country. You are unlikely to have allergies if you are from a family with a lot of kids, a large family size. You are unlikely to have allergies if you grew up rural and were around a lot of livestock. And if you were in an area with poor sanitation and a lot of Helminths. And so the idea with the hygiene hypothesis, I'm actually going to come back to this box in a second. Oops. So it could be because these are some other epidemiological sort of pieces of associations we've seen. That you are more likely to have particularly allergies if you were an only child, if you sort of were around in a sort of more urban lifestyle, if you had a particularly clean environment, and you in fact are statistically less likely to have allergies if you have older siblings. You are less likely to have allergies if you went to daycare. You are less likely to have allergies if you grew up on a farm. And so all of these things together kind of led to what the original hygiene hypothesis, or the way that the original hygiene hypothesis was described. And the idea with sort of the original hygiene hypothesis is that if you are in one of these situations where you have the small family or the westernized country, your immune system doesn't develop correctly because it doesn't see the right microbes. Because Th1 and Th2 responses are sort of interconnected and balanced between each other, because we got rid of all the Th2 pathogens and you don't ever see a Th2 pathogen in your life, your body starts making Th2 responses against wrong things, like pollen, or your lungs, or things like that. And so the idea is basically that because we specifically kind of removed these Th2 pathogens, you are now making some Th2 responses inappropriately. And so you can imagine, for example, if you were an only child, then it was probably, your less messy and gross around your house than if you had a whole bunch of kids, because your parents were, let's say you had a whole bunch of kids, your parents were like, whatever. Whereas if they're more likely to like be really on top of things with an only child. If you go to daycare, you're around all those other nasty kids. Whereas if you stay home by yourself, if you're around a farm, you might be getting more of these microbes. And so the idea is sort of this idea of, you know, microbial exposure is helping your immune system. There are lots of ways that that's made it into sort of pop culture. I will tell you that the idea is that, you know, in grad school we would like make jokes whenever someone would be like sneezing from allergies and be like, your mom didn't let you eat enough dirt. And it, you know, there's sort of this idea that is, I think you guys know, has sort of gotten into pop culture. I think that's part of the reason why I want to show you the sort of newer way we're thinking about it a little bit, because I think some of the stuff that people are doing in pop culture has gone way too far. But the idea was sort of that there was this issue in Th1, Th2 balance. We got rid of the Th2 pathogens. Now people are making weird Th2 responses across the board. And so that's sort of the original hygiene hypothesis.
So am I just crazy unfortunate that I have 3 older siblings and I went to all these different daycares and I did actually eat dirt. And I still have crazy allergies. I just have bad luck.
You may very well have bad luck. I will, and we'll say more about this when I talk about allergies next time. When I think about all of these things, I was an only child who grew up in a very rural. I was an only child, but I grew up very rural.
I thought you grew up in.
Yeah. There's unfortunately a lot of genetic stuff going on too, and I'm pretty sure in my case it's part of the genetic stuff. But so there are more things going on. But this is from your textbook, and this is sort of one of the alternatives that people have been thinking about with regard to the hygiene hypothesis. Because again, originally it was all sort of a deviation thing. We were all somehow getting our Th1, Th2 balance wrong. And some people said, well, are we sure that's what it is? Because when you are infected with a parasite, in particular a Helminth parasite, you are generally immunosuppressed. It suppresses your immune system a bit. And so if everybody in the population is infected with a parasite, then the whole population bell curve is going to be a bit lower. We're going to all as a population have a somewhat lower immune response than a population who didn't have parasites. And if our whole population has this lower bell curve, then only a small number of us are going to be at the really high level of immune responses that would lead to allergies. But if our whole population has a better immune system, has a stronger immune system because they didn't have parasites immunosuppressing them, then a larger fraction of us are going to be in this excess immune response category. And so maybe it's not about Th1, Th2 balance and all of these things. Maybe it's about sort of general immunosuppression going on. And so that was one idea that people started thinking about. Yep, Brenna.
I think I'm getting confused. Is a Helminth just a parasite? Or is it, like, are there different types of parasites? Or
So helminths are parasitic worms. There are different types of parasites.
Okay.
And most of the hygiene hypothesis stuff has been specifically studied with helminths.
Okay.
But depending on the literature, sometimes you'll see information about other parasites as well. Helminths are an example of a parasite. We often think about sort of how to think about this in parasites. I will also tell you that, especially because of the hygiene hypothesis, there have been some ideas out there that to deal with allergies, perhaps the thing to do is to make sure you make an appropriate Th2 response, is to make your body make an appropriate Th2 response. If the idea is really a problem with Th1, Th2 balance and you're not dealing with any Th2 pathogens appropriately, then make your body make a Th2 response appropriately. And so how would you do that? Yeah, Jonah.
Give your kid pollen and stuff.
Nope, don't give your kid pollen, because that would still be actually an inappropriate TH2 response. Nope, still an inappropriate TH2 response. Yeah.
Get parasites?
Huh?
Get parasites?
Yeah, get parasites. Purposely get a parasite infection.
Let's try it.
Yeah, you said you're hoping that was the end. And so people have actually done studies, not giving people actual parasite infections, but one of the things that they've tried to do is they've tried to give them pieces of schistosome eggs. So basically just take the like egg that can't reproduce, break it up and inject it, and see if just having those antigens around can cure the allergy. I definitely remember looking at a paper in grad school where someone was putting schistosome egg antigens in yogurt with the idea of trying to rebalance TH1 and TH2 responses.
Did it work?
So there were situations where it worked. It didn't always work. And so that perhaps is another reason why we can say, maybe this understanding of the hygiene hypothesis isn't totally correct.
Go out and get ringworm.
Huh?
Go out, get ringworm.
I would not recommend that you go out and get ringworm. But one of the ways that the hygiene hypothesis is now being talked about is with a different name. And so when I go to conferences and things, I'm starting to hear more and more discussion of something called the old friends hypothesis instead of the hygiene hypothesis. And with the old friends hypothesis, it's more of a thought about the fact that there are many microbes, Helminth certainly being one example, but in fact really there are many, that have been infecting people since people began. We co-evolved with microbes. We didn't co-evolve in a sterile environment and then all these microbes showed up. They were here, like 4 billion years before us. We have been evolving with them for a long time. There are many, many, many pieces of data that we have that say that our development, particularly our development of our immune system, is partially dependent on some microbes. And so maybe the idea is that we have all these things that are our old friends. We actually evolved in response and in collaboration with them. And so those things, whether they are bacteria, helminths, or other types of sort of environmental factors, may have resulted in us making a particular type of immune response. And more often we're thinking about the idea that perhaps our old friends induce a lot more of a Treg response. And so normally we should be making sort of this really nice Treg response. We should often be kind of getting our immune system being sort of chilled out. And you can see that could be direct, that could be indirect, where those old friends modify our immune system. But in any case, the moral of the story is we're ideally making a lot of Tregs. Now I want you to remember when we talked about helper T cell subsets, and we talked about cytokines that polarize helper T cell subsets. When we talked about polarizing a Treg, what did we talk about as the cytokine that polarized the Treg? So how did we make our naive CD4 T cell into a Treg in terms of cytokine polarization? TGF beta. When we talked about helper T cell polarization, did TGF beta show up at any other situation?
I remember my notes.
Yes.
It was in the lab.
You're picturing your notes and David says yes. So where did it also show up?
It was T helper 17.
So it was also involved with TH17 cells.
Right.
But what was the difference between when we would make it Th17 versus a Treg? Hmm? IL-6, it can also be something like IL-1 beta. Do you ever thought about those cytokines before? A few times? Where have you thought about them before? Huh? Where? They're part of the innate inflammatory response. They're sort of the key things that are made when you're under inflammation. You even measured them in the lab.
Full circle.
I know, totally full circle. And so maybe the idea is, originally, we should have been pushing a lot of our T cells towards Tregs, and maybe things have changed. Maybe we are now seeing more inflammation, and now all sorts of T cells that should have become Tregs and helped us be healthy are maybe getting pushed to Th17 because of inflammation happening at the same time. If we look at most of the increases in diseases that we see in a number of westernized countries, they are all diseases that have a chronic inflammation aspect. And chronic inflammation is being seen in many things. We realize that chronic inflammation, basically too much inflammation seems to be happening more and more often. This may be because of sort of genetics changing how we interact with microbes. Maybe it's that we're interacting with different microbes. We're not interacting with our old friends anymore. Maybe it's things like diet. There's a lot of literature about diet and inflammation and other things. All of those things may be leading us to suddenly have those inflammatory cytokines in addition to our TGF-beta, where we didn't previously. And that can start to lead to any number of different diseases, including a lot of allergies and autoimmune diseases. And so maybe the idea is, because we don't necessarily have the right relationship with our old friends, we're not getting this drive to Tregs in the way that we are supposed to. And maybe, in fact, we are even pushing sometimes more into a more inflammatory subset. Yep, Shelby.
I feel like I lost the plot a little bit.
Okay.
So, is that a response when it pushes towards Th17 when there is no parasite?
So the idea with the Th17 is that maybe there's something else going on in our lifestyle. Maybe it's either that we don't have the old friends. So imagine maybe we're making less TGF-beta, so we can't push the
So
Treg.
the response is to parasites or to something else?
To something else usually. And so this is sort of another view of all of this, and I adapted this very much. What should normally be happening is we should be chilling with our old friends. They should be helping us make Tregs, and those should kind of keep our immune responses chill. But because of lots of other stuff that may lead to chronic inflammation, there's going to be new kinds of PRR stimulation. We're going to get new inflammatory cytokine production. Maybe this will help, Shelby. If you look at how I wrote this, I wrote like stuff that gives us cytokines, because it could be a Th2 or it could be a Th17 or it could be anything that's not a Treg.
That's being produced.
That's being produced and pushing you away from Tregs.
Okay.
So I wrote it very vaguely here. That you could, because of sort of other sort of lifestyle changes, we could be pushing our immune system towards things that are not Tregs. Whether those are Th1 or Th17 or Th2 kind of depends on the disease and what the stimulus is and what's going on. And so maybe this is actually about we're not making Tregs well because we don't have our old friends, and we're making too much of other stuff because we have things in our life that are not great. And so you can see there are a lot of diseases where we think that the idea might be this diminished induction of Tregs. And we actually have good evidence that it's something about some reason why we're seeing that reduced number of Tregs is related to our microbial environment. For a lot of autoimmune diseases, things like inflammatory bowel diseases and allergies, there are other disorders where people have hypothesized that microbes might be part of it, but it's not really clear. And I will tell you, when I was putting this together, I found a lot of literature that I was trying to use that all came from this one scientist, Rook. And some of his stuff is really good, and some of it I was like, ooh, you just went 27 steps further than I think you should have. I was like, whoa, let's pull it back a little bit. The stuff on the top I think is relatively certain, but I think that there are other things that are much more speculative. Let's see what's out here. Yes, Jonah.
So, I know with like a number of those diseases, the drugs related to them are mostly like anti-inflammatory drugs. Is the point of that to inhibit that and to try to increase Tregs, or is it just to?
Well, I'm gonna get there. I hope I'm gonna get there. But before I get there, I want to tell you some other things about autoimmunity. And so remember that big picture, we're thinking about autoimmunity as resulting from a failure of self-tolerance. And we've seen a lot of examples of sort of environmental exposures or infection that may lead to changes in immune regulation and autoimmunity. But what I hope you notice here as well is that in both of these images, there's also a role for individual genetics. Everyone who is who goes through some of these particular environmental exposures does not end up with autoimmunity. I did a podcast yesterday about a really cool paper that was getting at both parts of this, in fact. And so people have done a lot of genetics studies where they have been looking at genes associated with autoimmunity. And in fact, in some cases with allergy as well. Some of these are shown as allergy. And so here you see all of the human chromosomes, and you see different little shapes and colors indicating different places in the human genome that have been statistically associated with one of these different diseases. You can see that there are associations throughout the genome. You can also see that there are some genes, like this sort of short arm of chromosome 20, some areas, that are related to lots of autoimmune diseases. You can also see that there are some associations that are specific for only like one particular autoimmune disease. And so some part of this is thinking about aspects of genetics. When we think about sort of the strength of the statistical association, where the association is biggest, there's one gene that really stands out as the gene that is related to autoimmunity. Sometimes I will see people spend like extremely large amounts of money to do these huge genetic studies, and they come up with the one gene that is associated with this disease is MHC, and I will say, thank you, Captain Obvious. And so really the gene that is most closely related or most closely linked to autoimmunity is MHC type. And so you can see, for example, in ankylosing spondylitis, most patients have HLA-B27. So in fact, more than 95% of patients with this disease have HLA-B27, while it's much more rare in control patients. And so we can also see a relative risk here. You can see birdshot chorioretinopathy. Patients with that often have HLA-A29. And you can see a lot of different things here. You can also see there are some sort of negative associations, like type 1 diabetes patients are unlikely to have HLA-DQ6. the basic idea with all of this is that when we are going to have some kind of reaction to self-antigen, that self-antigen isn't presented on every kind of MHC. And so if there's one self-antigen, there's a self-antigen in some virus that overlaps with some self-antigen that's going to lead you to have some autoimmune disease, everybody who gets infected with that virus isn't going to make a response to that mimic epitope. Only the people with the right MHC molecule to present that peptide are going to make it. And so usually any particular self-peptide can only be presented by some MHC types and not others. And so statistically, we see that certain MHC types tend to be very much associated with certain diseases, with the idea that the self-antigen is only presented by some MHC type. And so you have to have that MHC type. So now we're thinking about you got to be unlucky enough to have that MHC type and to have that particular infection on that particular day with the naive T cell. So you can see it's a lot of sort of questions of luck here. There are some other genes that are associated with autoimmunity. And so you can see some of those disease associations here. So you can see a bunch of different autoimmune diseases listed here, and you'll notice, for example, one big group of disease associations or polymorphisms in particular genes associated with those diseases are in cytokines, cytokine receptors, cytokine signaling. Not a shock to imagine how cytokines could be involved in autoimmunity. Some of them are involved in innate immune responses. Some of them are involved in co-stimulation. Some are involved in antigen presentation. And so we have a lot of sort of other genes that are associated with immune regulation that can be part of autoimmune disease. There's sort of another set of genes that are not really shown here that are also potentially involved in some autoimmune diseases, though this is also a little bit more of an allergy thing. So sometimes, for example, if you have an autoimmune disease that particularly impacts your skin, say, you might have a polymorphism in a gene that changes like a skin protein. And so you also might have something unique to the organ that is being attacked that may be seen as different. And so these are some big genes associated with autoimmune diseases. But the other thing, of course, that we've already talked about a little bit is that there is a pretty major association between sex and autoimmunity. And so if we look at many autoimmune diseases, we see that those autoimmune diseases are seen much more frequently in women than in men. And so you can see, this is a figure from your textbook on the left that shows how many times more likely something is in men or in women. If it's pink, it's in women and it's going to the right. If it's blue and going to the left, it's in men. If it's sort of purple, it's like in between. And so you can see like Addison's disease, they're showing as more than 12 times more frequent in women. Sjogren's syndrome, more than 12 times more likely. Lupus, you can see lots and lots of different disease states. And I've got a table over here as well. So you can see Hashimoto's thyroiditis, 19 times as many women as men. Sjogren's, 16 times. Sclerosis, 12. Lupus, 9. Graves' disease, 7. And so there is this massive overrepresentation of many of these autoimmune diseases in women. As I've mentioned to you before, one hypothesis for this has related to gene dose. There are a number of important immune genes on the X chromosome. And so women have 2 copies, men have 1 copy. And in fact, these genes do not always get inactivated, like other genes on the X chromosome. So women really do have like 2 times as much of the protein. So that may be part of it. As we talked about a lot in lab, hormone exposure may be part of it. The big hormones that we talked about, estrogen and testosterone, are sort of part of a steroid hormone group. Steroid hormones are basically lipids. They go straight through the cell membrane and just impact transcription. They don't really need much signaling. And we seem to be seeing more and more data that things like estrogen, testosterone, those steroid hormones, impact immune responses a lot. One of the other things that people have talked about in relation to this is that we often tend to see these autoimmune diseases. In general, autoimmune diseases show up sort of a little bit later in life. And so that's one thing that's sort of interesting in terms of, natural selection. They're generally showing up, when people have already had some time to survive and reproduce. So it may be hard for there to be selection against them. They may be a consequence of us having a very broad immune response. But in particular, the autoimmune diseases that I'm showing here tend to show up in women after their first pregnancy. And so one other idea, and we certainly know this, that pregnancy dramatically changes the immune system, because in fact you basically have to not reject a foreign fetus for 9 months. And so your immune system has to change really, really, really dramatically and do some really crazy things. And so it may also be because of all of those changes that may happen during pregnancy that may predispose one to autoimmunity. And sort of how this is just late in like a timing thing. Remember that I told you before with autoimmunity that there's sort of this idea of there's the thing that happens that breaks the tolerance, and then you don't end up in the doctor's office like the next day. You end up in the doctor's office many years later, because it takes a while for that T cell, to go destroy whatever before you actually notice symptoms. And so part of this might just be you're not going to notice this till you're old because you need some time for the destruction to happen, and how much that's related to other kind of life timing things is an interesting question. All right, so usually at this point, I start talking about some specific details of specific autoimmune diseases. I think I'm going to switch some stuff up and do some things in a different order right now. There's one thing I want to mention that I would get into with regard to some of these autoimmune diseases before I do my little switchgear thing. And that is that when we talk about autoimmune diseases, we often talk about them as being either mediated by T cells or mediated by antibodies. We usually talk about a particular type of immune response that is really important for those autoimmune diseases. And that is largely true, but as your textbook shows, I'm going to tell you certain diseases are more T-cell mediated, certain diseases are more antibody mediated. It's not like it's 100% T-cells and 0% antibodies for a T-cell mediated disease. Often there are a lot of different parts of the immune system that are coming together to play a role here. And the reason why I'm going to, I usually talk about this, and why I'm going to, I think, sort of scale a little bit of this back, is that, like I said, we often think about our autoimmune diseases as being T-cell mediated or antibody mediated, and we can think about specific mechanisms. The next topic we're going to talk about after autoimmunity is allergy, otherwise known as hypersensitivity. After we talk about allergy, we're going to talk about transplantation. it turns out that all of these things have a bunch of stuff in common: allergy, transplantation, and autoimmunity. And one of the things that they have in common is that we see similar mechanisms. So there's going to be one situation where T cells are usually the problem. There's going to be one situation where antibodies are usually the problem. We can actually divide up the reactions that we're seeing, or the types of responses, into a type 1, type 2, type 3, and type 4 hypersensitivity response. And so I can put certain autoimmune diseases in these groups of type 1, type 2, type 3, and type 4 mechanisms. But when we start doing allergies, I'm gonna give you examples of type 1, type 2, type 3, and type 4. And when we do transplantation, I'm gonna give you examples of type 2, type 3, and type 4. And I would be doing the same thing when I talk about autoimmunity. And so you're going to get to know these mechanisms pretty well in some of the other settings. So what I'm gonna do is I'm actually gonna skip to sort of some of the stuff I put at the end of this lecture to make sure I have time to talk about it, because I think it will be fruitful for us to have in the discussion. And depending on what time I have left, I may come back to some of these specific examples, but I want to hit some of the other stuff. I want to be sure I have time for some of the other stuff. And that other stuff is very much related to Jonah's question about treatment of autoimmune disease. And thinking about treatment is also something that we're going to see kind of over and over again with all of these mechanisms. And for us thinking about sort of immunology in the future and where immunology is going in the future, I think this is an important thing to think about. So if you think about treating autoimmunity, my family has a bunch of autoimmune diseases, so I have sort of known about details of autoimmunity and autoimmune disease treatment, since like the 80s. And if we think back through most of that time, 80s and 90s, there was kind of one option for treating autoimmune disease. And that option is still an option today. You probably know of people who have been treated with this, or know of situations where this treatment has been used. I will also say some of the treatments that we're going to talk about right now with autoimmunity end up overlapping into allergy and transplantation because it's similar mechanisms. But I think that I want us to talk through some of the treatment issues now. And so the first kind of big treatment that was an option was treatment with steroids, particularly treatment with either hydrocortisone or prednisone. And again, you might be able to think of situations where you've heard of those for allergies or transplantation as well. Sort of all kind of the same idea. These are steroids. They are small molecules that are lipids that you can see their structures here. They diffuse directly across the plasma membrane. They impact transcription quite a bit. And they impact transcription in a lot of leukocytes. So it's not just one kind of leukocyte. They're basically impacting all the kinds of leukocytes. And they're impacting them really broadly. So you can see they will decrease the production of a whole bunch of cytokines. They'll decrease adhesion molecules. They'll decrease a whole bunch of other things. And so we're just going to, if you think about it, kind of depress everybody. Just turn down all the leukocytes. Just make everybody's transcription come down a bit with this steroid treatment. One thing that's sort of interesting, oh my gosh, these are like these corticosteroids that we've used for I don't even know how many years as sort of the thing to chill out inflammation, the thing to chill out your immune response, are in a chemical class that's similar to things like testosterone and estrogen. Maybe it shouldn't surprise us that testosterone and estrogen impacts cells pretty broadly. These are also in the same chemical class as cortisol. What's cortisol? Stress hormone. It's the hormone that you make when you're under a lot of stress. these steroids we're basically using to just depress your immune system, to shut your immune system down. Hey, wait a minute, maybe cortisol, maybe being stressed just dampens your immune system and shuts your immune system down by the same mechanism. Across all leukocytes, which does seem to potentially be the case. And so, stress, not great for your immune system. But let's think for a second about exactly what's going on here with this steroid therapy. When we are treating a patient who has whatever autoimmune issue with a steroid, we are basically, well, we can imagine, giving this drug to basically every lymphocyte or every leukocyte in the whole body and turning it down. Can you see any problems with that scheme? Yeah, Brooke.
More susceptible to other infections?
Yeah, more susceptible to other infections. You have basically just taken the entire immune system and turned it down. And so you're going to be more susceptible to other infections. You might have a lot of other kinds of bad effects because you have turned everything down. In some ways, you can think of this as like a little bit of a sledgehammer approach, right? And in a lot of biomedical science, we have situations where we have treatments that are kind of sledgehammers, that can turn things down in like a really crude way. And so we'll talk about this more later, but think about like cancer. We don't necessarily have a great way to kill the cancer cells, we just kill any cell that divides too quickly. And that's why you also, like, when you have chemo, lose your hair and have GI issues, because we killed so many cells. We can't be very specific. And here, we're not being very specific. We're just turning down the entire immune system. And you can see some reasons why that might be problematic. And so over time, the thing that immunologists have done is they've come up with new treatments. Here I'm going to tell you about some of these new treatments for autoimmunity, but you will see similar themes as we talk about treatments for other things. And the big picture idea with these treatments is that in every case we're trying to get more specific. We're trying to not sledgehammer the whole immune system, but maybe try to impact only a smaller portion of the immune system. So we're not making you immune compromised across the board. We're trying to make this more specific to just stopping the immune response that you have that's a problem. This table shows you some of the many ways that people are doing this. This is a huge deal in the pharma industry. What you can notice, sort of as your spoiler, is that sometimes we try to get rid of a particular cell type, and sometimes we actually target a specific step that we as immunologists have learned about, and that we as immunologists have realized is key, and try to just stop that step without stopping the entire big picture of the immune response. And so one of the first things that people actually started to use was cyclosporin A. And so cyclosporin A is used in transplant, but it's also used in a number of situations where we have autoimmunity. And so cyclosporin A and some similar types of drugs act by, if you recall, getting in between calmodulin and calcineurin so that we can't have calcium activation happen in T cells, and so that NFAT can never be activated, we never actually turn on our T cell. So this stops T cell activation. So how does this improve upon steroid therapy? Yeah, Josh.
You're targeting the T cells rather than targeting the whole immune system.
Yeah, so now we're turning off T cells, but we're leaving B cells and all of the innate immune cells to work. So we've sort of narrowed in on what we're inhibiting. Can you see any problems with cyclosporin A treatment? Yeah, Brenna.
So you need the B cells to work. They might be able to work without the T cells.
So A, it's probably not going to be super helpful for the B cells if the T cells aren't working. So that's one issue. Any other issues? Yeah, David.
You're also not just impacting the problematic T cells, but also the helpful ones.
You're still impacting all the T cells. And so you're still actually going to have a patient who's pretty immune compromised. If they can't deal with any microbe via T cells, yeah, you're going to stop their disease, and you're not going to be quite as severe as treating them with a steroid, but you're still going to be a pretty, I don't know, it's a skinnier sledgehammer. And so what we really want to keep doing is thinking about ways that we can kind of narrow this down and really try to hit just the pathogenic cells. We want to be specific with our treatment. Have you heard me use the word specific or specificity earlier in this semester? Yeah. When did I talk a lot about specificity? Huh?
Adaptive.
I told you that's the whole point of the adaptive immune system. You have this entire body system that has evolved to be really, really specific. So can we as biomedical scientists take some lessons from the specificity of the adaptive immune system and use that in our treatments? And that's a big thing that we're doing now. If you are to think about what, sort of, again, where the pharma industry is going, it's going into things like this, whether it's for autoimmunity or allergy or transplantation or or cancer or a million other diseases that are not like this. The key is, wait a minute, we know about specificity in the immune system. So let's see if we can use that knowledge of specificity to make our treatments better. And most commonly what we will do is we will design antibodies that can specifically hit one particular type of molecule. So again, we're getting more specific by using the antibodies. And the idea is that we are using the antibodies because we can make the antibodies in the lab and deliver them to a patient like it's a small molecule, like it's some kind of medicine. But we're using a thing that's really specific. And so the first idea for using antibodies to be specific and to deal with autoimmune diseases was to use antibodies like zanulinimab or rituximab, which is sort of one of the really famous ones. These are both used in rheumatoid arthritis, and they do things like binding to either CD4, which are all CD4 T cells, or rituximab, which is a pretty famous one, binds to CD20, which is on activated B cells. And when these antibodies bind to their target, so here you can see an activated B cell, it's getting bound by rituximab, that leads to that B cell getting destroyed, getting depleted or deleted, either because it's being lysed by an NK cell, it's being killed by complement, whatever. So basically, now we're going to destroy all of the B cells that have CD20 on their surface. How is this an improvement over what we saw before? Yeah, Jonah.
Instead of reducing generally T cells and in that case B cells, it's now just targeting, I guess, a specific set of B cells.
Yeah, so now we're targeting B cells, and we're only targeting B cells that are activated, which the ones that are perhaps making antibodies that are giving you an autoimmune disease are activated. And so we're getting rid of all the activated B cells. So again, we're getting narrower. And rituximab is used in a bunch of different situations. It's really, really helpful for many patients. But we're still not, super narrow. Our patient's still going to be immunocompromised because we just got rid of all their activated B cells. But it's more narrow than the previous treatments. Similarly, people have come up with a molecule that is CTLA-4 Usually what they do is they put CTLA-4 on an immunoglobulin domain, so it looks like it's an antibody, but it's actually CTLA-4. And so you can see this CTLA-4, abatacept or Orencia, that's used for a number of different disease states. And so if we have a lot of CTLA-4 in the body, it will bind to all the CD28, and now T cells can't get turned on. So we can't activate a T cell because we've blocked the CD28 B7 interaction. And so you can see this happening here as well. Here is a T cell that is potentially going to interact, make a self-reactive response. Now we are going to use this molecule that binds up all the B7. We can never give signal 2, and so we're going to in fact energize our T cells that are seeing self-antigen or seeing antigen instead of turning on our T cells. People who described this is important in a number of different disease states, and people who described this medication won the Nobel Prize, I think it was 2015, around. So again, we're narrowing in a bit, but we're still kind of broad. But again, this is something that has been revolutionary for a lot of patients. The big place where a lot of these kind of specific autoimmune therapies have been in thinking about cytokines and cytokine-directed therapies. And so this is an incredibly out-of-date and short list of some of the cytokine-directed therapies there are. There are so many now. Where basically what we have is we have some antibody that binds to a particular cytokine and blocks that cytokine. So that cytokine can no longer interact with cells. So no matter how much of that cytokine you make, you're never going to see the response of that cytokine. And you can see that there are versions that involve TNF. This shows some that involve the interferons and IL-1, IL-17, IL-23, IL-13, IL-4. Like so many are possible now. Literally, when I watch TV, I cannot watch TV without seeing an ad for some cytokine-directed antibody. And I know it's a cytokine-directed antibody, but like most people are not necessarily going to know. And so you can see kind of how this might work. Normally, TNF might bind to the TNF receptor, but if we treat with an antibody, the antibody is going to bind to all the TNF, and that's not going to bind to the cell. Or we can have a situation where maybe we bind to the receptor and keep the cytokine from binding to the receptor. And so the basic idea is we're blocking some kind of cytokine receptor interaction, either by soaking up all the cytokine or by blocking the receptor. And this also shows people have figured out ways to make not just a whole antibody, but can they make just an FAB or something like that, because it's smaller and can get in little tight places between the cells. And so the idea is that now we're blocking these cytokines. And so now, instead of blocking, say, all T cells, maybe we're blocking just Th17 cells, but you can still make Th1, Th2, and Tregs. Or maybe we're blocking, one particular type of inflammation while leaving all the others intact. So we're getting more narrow, but we're still not just hitting the pathogenic cells. We're still, potentially blocking a lot of cells. We're still immunocompromising our patients. We've done way better. Just to give you an idea, these are graphs. The blue lines are showing kind of levels of things in a rheumatoid arthritis patient. The red is showing what happens with patients who get anti-TNF. I can tell you from experience of members of my own family, life-changing in terms of these drugs, but those people are still somewhat immunosuppressed. They still can make no TNF-related immune responses. And so these drugs have been amazing. We have therapies that basically can block signal 1, signal 2, and signal 3 on T cells, and we can sort of specifically hit whichever part of a T cell receptor signaling we want and try to really impact T cell responses a lot. One other thing I want to say before I sort of end on this is that one of the things that you notice is that with many of these therapies, we're blocking sort of the end effector, like the cytokine that's causing the damage, right? We're not doing something like making it so you never break tolerance in the first place. There's sort of that moment where you break tolerance in the first place, and then many years later you have enough damage from an immune response that's causing severe issues. We don't, and again, we're only kind of making drugs that are hitting that very end stage. We're not making drugs that are hitting that beginning stage. So in fact, I can block all the TNF I want. My family member is still going to have autoreactive T cells that are still going to be trying to respond. At the same time, and so sometimes people are like, I want to block the autoreactive T cell from ever being formed. Well, A, we don't really know how that works, so it's hard to do that right now. And B, how do you figure out that you don't even show up at the doctor at that point? Because you don't necessarily say, oh, I think I had my 1 T cell break tolerance today. And so how you would actually treat that is really unclear. But what I want to point out, and this will be, again, particularly important with, say, like transplantation, with diabetes, we know that we have certain T cells that are destroying parts of the pancreas. And people come up with all these great ways to, like, replace the pancreas that got destroyed with transplantation. Well, you still have the T cells. You're going to destroy the replaced one too. Unless you do something about the T cells, you're just going to keep killing pancreases, no matter how many times you put them in. So also think a little bit about whether a treatment and whether a strategy is getting at sort of that initiation of the disease versus the effector part of the disease. That's something we'll see as well when we think about allergies, which we'll start thinking about on Friday. And I will see you guys in lab and send you an email when I have the lab handout posted.